Forensic Biology. Welcome to class today. We will be looking at the first part of Chapter 11, Entomology. So today we will cover Part 1, going over introduction, how forensic entomology is used, and some of the history of forensic entomology. So let's go ahead and get started. Okay, here are some of your objectives. They're on here for you. Describe several examples of the ways the forensic entomology is used to solve crimes. Compare and contrast 4 stages of blowfly metamorphosis and describe the significance of blowflies in forensic entomology. Describe the function of each of the following organs on blowflies and explain the significance of each structure to forensic entomologists: spiracles, mouth hooks, crop. Describe the effect of different environmental factors on insect development. Describe the 5 stages of decomposition. Relate the process of insect succession to the changing environment that occurs during the stages of decomposition. Explain how forensic entomologists interpret forensic evidence and environmental conditions to estimate a PMI. Explain how insect evidence is analyzed to provide evidence of the deceased person, identify person's identity or drug, poison, or toxin exposure. Summarize the procedures for documenting and collecting insect evidence from a crime scene. So once again, those are our outcomes. That's what we want to be able to do at the end of this chapter. Not too many of those in this lecture. We will be looking at a little bit more. Here's your vocab. Following along in the book, these are the terms that are bold in your book. You will need to know those. So let's jump into it. Introduction. So we are going to be talking about insects. So going back a little bit, we're going to be looking at a phyla, which is a category of animals. If we're looking at Carl Linnau's classification type systematics, you have kingdom, okay, Animalia, and then you have phylum after that. It's just a way of grouping animals. Well, we're going to be looking at arthropods. Arthropods are pretty much insects, spiders, octopus, crustaceans, things like that. That is going to be pretty much our look into animals, and it's going to be similar to that of what we looked at in chapter 12 with the decomposition and things like that, but now we're applying it to insects for the most part rather than humans. So an insect, the reason why we are looking at insects, there's a lot of them. They're attracted to different things, so you can get a lot of information based off of that. So going on, an insect's going to have 3 body segments. Going to have a head, it's going to have a thorax. Now the thorax is going to have the legs. The legs are going to come out of the thorax, and then an abdomen. Now most of these insects fly. Okay, not everyone, but most of them are going to have some sort of way to fly. There's said to be about 6 million different species of arthropods. Your book says about 1 million, but everything I've looked at is the lowest number I've seen is 6 million. And they make up the majority of the animal kingdom compared to any other animal. And pretty much what we're going to be looking at is the not necessarily decomposition, but the stages of these animals, because they go through certain stages. So it helps you to be able to create a PMI. Okay. So going into it, this is that stages that we are talking about. Okay, so they're going to look into civil and criminal investigations, usually of neglect or criminal, homicide, things like that, a dead body, to determine kind of how long it's been there. So that is going to give a PMI based on these kind of different succession is what it's called. So eggs, okay, obviously here you have blowfly eggs. Now blowflies are going to be the insect that we're going to look at the most, as it's going to be kind of the framework for all of this. And I'll explain a little bit when we get to history. But pretty much what happens is you have eggs, then you get hatched, you're a larva, then larva go into pupae, then nymphs, then adults. So you have these different stages here, and throughout this whole stage from eggs to adults is called a complete metamorphosis, simply because you are changing from an egg all the way to an adult. So if we're looking at a fly, you're looking at an egg to a larva to something that you look and consider as a fly. So it's just a complete change as far as what's going to happen there. So the job of the forensic entomologist is to collect and identify insect evidence and interpret it in relation to environmental conditions and variables that exist at crime scenes. So pretty much what's going to happen is they're going to collect the evidence, usually found on the body or around the body, and then they're going to record the evidence of what's called ambient environment. That's going to be kind of the temperature, humidity, stuff like that, because what you want to do is recreate that environment in order to conduct your own experiments to see how long how far along the life stages these animals or these insects are. So you're creating a time frame as far as how long is it for that. So that's pretty much what a forensic entomologist is going to do. They also will be working with toxicology, botany, and autopsy to get a full-rounded evidence and analyze it. So they're not just alone, but you can do a lot just by themselves. But most of the time, we'll talk about it. You can get a lot of information from them based on toxicology, plants. So there's a lot of different stuff that work together, but an entomologist is simply going to be looking at insect stages and creating a PMI in that regard. So how's it going to be used? Okay, so there's different ways it can be used. One, locate a body. I will talk about an example in the history section of this where you can locate a body or flesh or blood or anything like that. You can see flies and things like that. There's something that is decomposing over there that's getting eaten, but also determine if the body was moved. Okay, now there is one thing I do want to talk about here is it can get a little construed. Say if you have a primary crime scene about a homicide that happened inside of a house, then that person dies inside the house and then later gets moved out to the forest. That's going to help. That's going to hurt with determining the actual PMI of that. But you can use the things we learned from chapter 12 and the decomposition of the body along with the entomology to get the best time frame or the best PMI available, but it will determine if the body's been moved or not based on one, the local area and their insects and things that are local to that, as well as a matching PMI between the body and the insects that are going to be used. Can identify geographic range of the crime, once again comparing the kind of the profile of the insects that are found on the body to the surrounding area. If it matches, it's local. If it doesn't match, then it came from somewhere else. Link a suspect to the crime, evidence of bite marks, things like that from a suspect that was at the crime scene. Indicate whether it's been traveling, and also trace origin or drugs as well. So pretty much what you are what you eat. Well, same thing for animals. They eat or bite into flesh, things like that. You can trace the drugs. Also, a lot of times you find insects in drugs or in, they get into everything, flour and sugar and stuff like that. So make sure, you have a plastic bag and things that animals can't get into when you are storing those. Okay. Like I said, limits for entomology. It can get kind of squirrely with if they're killed in multiple or inside where there are no bugs to outside where there are bugs. That can ruin the time or the PMI, but you just have to work it with the body decomposition as well to create that PMI, and if they match up, then they were killed at this spot. If they don't match up, then there's another crime scene. Okay, so what questions are they going to answer? Where the body injury is at. Usually you'll see that with maggots. Maggots like a warm, moist environment. So if there's an injury that occurs and exposes the inside, the maggots usually get laid in there, and they're going to be able to develop and eat kind of the nutrients from the body. Was the body moved after death? Victim restrained while alive. You'll see restraint marks. A lot of times you see what this is looking for is neglect, because what happens is when you leave somebody alone, okay, for an amount of time, your body has certain things that have to happen, such as urination, defecation. Those smells and things like that are going to attract certain types of animals, or insects for the most part. So depending on what type of insects are there will help determine whether that person was restrained while alive. And then also whether it's covered, buried, submerged, or in water, based on, once again, the type of animals. Was it deceased, exposed to any toxic chemical, or under influence of any recreational drugs? As insects kind of chew away at you, you're able to receive a toxicology based on their structures that they have. So you can understand if they were, drunk, or they had alcohol, or they had drugs, or anything like that. So quite a bit that bugs can tell you about what happens when you die. So a lot of information. So here are some examples as far as some history. You will need to know this. This will be our last slide for this part 1. All right, so the example of determining dead bodies using forensic entomology to determine or locate dead bodies is an example here in 1247, okay, which was published in the Washing Away of Wrongs. This was from Sung Soo, and pretty much, or Sung Soo is the one that published it. And what happened was a bloody murder was discovered by a group of farmers returning home from their fields. All the workers were instructed to lay down their sickles, their utensil to cut. And then soon flies identified the murder by landing on the one sickle that contained microscopic remnants of flesh and blood. So the murderer was, or at least the murder weapon was identified because it had blood and flesh on it, which attracted the bugs. So that's one example of how you can find and locate either a body or a weapon or something like that, as that flesh is going to attract the bugs. In 1855, insect evidence was used to solve the death of a murdered infant hidden in a wall. So a little bit more about this is you have French entomologist Bergeret d'Arbois. I think I was close. So pretty much what happens is they found a mummified, murdered infant that was pretty much stuffed in a wall. And what they do is they take what is called insect succession. Not only insects go through these stages of metamorphosis, but you also have at different points in time certain animals or certain insects are going to be attracted to that body based on decomposition and things like that. So what he does is he goes through and he understands that there's a process, and he creates a PMI. He links it back to the owner. The house had gone through about 4 different owners at the time, so creating that PMI, linking it to the owner, and then eventually arresting the owners for the murder of the infant. Then in the early part of the 20th century, research done in the effect of screwworms had on living cattle and sheep laid groundwork for future entomologists. Pretty much what is happening here is they create what was called the blowflies of North America. Is they took these flies, and they went through these successions and developed these procedures as far as standards. Pretty much that's what happened, is they made standards. And not only that, but they're going to go on to what is called the Dober standard. And pretty much what they do with that is, not only do they have the standards with the stages of insects, but also with the correct environment as well. As an environment, it's going to have an effect on speeding up or slowing down the process. So the Dober standard is not only just the stages, but also the environment as well. And that's what is used in court nowadays. It's called the Dober standard, and that was developed in the University of Tennessee. But Dr. David G. Hall, he's the one that wrote the Blowflies of North America, and that's pretty much going to be the Bible, if you say, for forensic entomology. And pretty much what it does is just going to lay the groundwork and standards for the different stages of insect succession and the different stages of metamorphosis for blowflies. So that's part one. Next time we will cover insect and decomposition. There's quite a bit. We'll look at decomposition. We'll be taking a look at bottle flies or blowflies, same thing. We'll be looking at morphology, body shape, house flies, flesh flies, coffin flies, and beetles and other insects on decomposition. This section is going to be about 3 parts, and then your quiz will be the following week. So after part 3, so the next day after that would be your quiz, and that's pretty much it. I think we've covered it all. Maybe one more section if we got time for that. So thanks for tuning in, and enjoy